Today we are going to be learning about microorganisms, specifically bacteria.
Bacteria and people. What is a bacteria? Bacteria is a prokaryote, which is a single-celled organism. Humans are made of 90% bacteria, which means that you are only 10% you. Most of those bacteria are harmless, and most of them are actually good for you, but some of them are bad. Those are called pathogens. Those are bad bacteria.
Counting and classifying bacteria. Currently on the planet, there is an estimated 5 nonillion bacteria, which is a 5 followed by 30 zeros. There are thousands of bacteria types, but the three main ones are Bacilli, Kochi, and Spirulli. All three of them are in this picture. If you do the math, you have around 7.14 vigintillion bacterium in your body alone.
Some positives to bacteria would be that it helps to clean up oil spills. It helps with gene therapy, which is pretty much just taking a mutated gene, you take it out, and then you put in a new and functioning gene to make a functioning cell. And that also helps to kill pesticides, which you don't want. But the most important use of bacteria is that it helps with digestion. You need bacteria to eat food, and without it, we would all die.
Some negatives to bacteria would be that it causes sicknesses, it breaks down skin and forms rashes, and they are transmitted by vectors like ticks, which carries Lyme's disease, which is a bacteria. A vector is a carrier of bacteria. As mentioned before, ticks can carry Lyme's disease from deer to humans.
Controlling bacteria. Most bacteria can be killed at 71 degrees Fahrenheit, but you might need antibiotic drugs to get rid of them if they don't die. These are mainly used in humans for vaccines to kill off bacteria, but some of the bacteria can have resistance to that drug, and they survive the effects of the drugs in them. On the picture, the white bacteria have resistance, and the black ones have no resistance, so they die off while the others survive.
Gram-positive and gram-negative bacteria. Different types of bacteria stain different colors when gram stain is applied. Gram staining is where a dye is added and then removed, resulting in either a purple or a pink stain on the bacteria. The differences between gram-negative and gram-positive is that gram-negative has a cell membrane and a thin cell wall, while gram-positive has no cell membrane and has a thick cell wall.